everyone, and welcome to today's webinar titled Walk Away from the Laborious NGS Library Prep Introducing AVENIO Edge System. This webinar is a part of the 6th Annual Laboratory Automation Virtual Event. I am Antonina Salcido of LabRoots, and I will be your moderator for today's event, with help from Andy Chang of Roche Sequencing. Today's educational web seminar is presented by LabRoots and sponsored by Roche Sequencing. For more information about Roche Sequencing, please go to sequencing.roche.com. Now let's get started. Before we begin, I'd like to remind everyone that this event is interactive. We encourage you to first participate by communicating with other attendees using our new live chat feature during the presentation. You can find the live chat located at the right of your screen. You can also participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Submit. If you have trouble seeing or hearing the presentation, click on the Help Desk button located at the bottom of your screen within the navigation bar or from the lobby. I now present today's speakers: Teri Mueller, Field Applications Consultant at Roche Sequencing and Life Science, Scott Verreau, Senior Automation Support Scientist at Roche, and Andy Chang, Marketing Manager at Roche. For a complete biography on our speakers, please visit the Biography tab at the left of your screen. Teri, Scott, and Andy, you may now begin your presentation.
Hello and welcome. Thank you for joining us today. My name is Andy Chang. I'm the Marketing Manager supporting NGS automation at Roche Sequencing and Life Science. As many of you know, library preparation is an important and essential part of NGS workflow. But no matter how streamlined the library prep protocol is, it is just not fun to do. It's laborious, time-consuming, and it requires a lot of focus to not make any mistakes. So that's why lab automation has become a great solution for NGS library prep. But automation also comes with its own challenges as well. Learning how to use a system, developing new protocols, keeping up with all the hardware, software updates, and maintaining the equipment just requires a lot of effort and time. Today, we have invited two of our Roche NGS experts to help us uncover some of these challenges. We will also take the opportunity to share more about our new AVENIO Edge system and explain how it is designed to overcome some of these challenges. But first, please let me introduce our NGS experts, Teri and Scott. Teri Mueller is our Field Application Consultant who has been with Roche for over 15 years. Teri has various NGS applications and instrumentation experience and has spent over 23 years in the field of genomics. Scott Verreau is our Senior Automation Support Scientist who has spent over 20 years working in the field of NGS. He has developed NGS workflows and automating them on various automated liquid handling systems. It's great to have both Scott and Teri to join us today. Scott and Teri, in your careers, you have supported many NGS applications. What are some challenges you have seen, and can you share some of your personal experiences with us as well?
Yeah, thanks so much, Andy. I would say easily it's the amount of hands-on time that's needed. You know, a lab tech is basically glued to their bench for 2 days or more with only small windows of time to eat lunch or even go to the restroom.
Yeah, I totally agree. That's why I learned how to program liquid handlers. If more people would invest in automation, they'd quickly see that they can process a lot more samples, the same amount of people, which would allow those people to go and work on other things.
Yeah, but to get automation up and running, you need someone who not only knows how to program the liquid handler, but also understands the biochemistry, right? So I seldomly come across a lab person with both of those skills. So now you have to have 2 people devoted to that. So not to mention, you also don't just need the liquid handler, you also need a thermocycler, a magnet for the cleanups, the shakers, and then enough space to fit all the plates and tips too.
All true. And this is the main thing that people underestimate when they're trying to build a process around automation. They think they can just go out, buy all the components, put them together, and it'll just work. But in reality, it can take half a year or more to finalize one of these workflows. So, Teri, besides reducing hands-on time, what else have you seen in the field that's a pain point for customers trying to do these manual processes?
So I would say the next one would be just people. There are a lot of labs that are understaffed for the amount of work that they're expected to do. And because everyone is running around trying to get so much done, it's easy for technicians to get interrupted when they're making a master mix or pipetting into a plate, which can, of course, then ruin those samples. And then you can see quite a bit of variations between technicians. So one might like to tip mix versus vortexing, and the amount of time that they do that. So that can cause a lot of variability within the data.
Yeah. Again, this is the great thing about liquid handlers. They can only do what they're programmed to do. Transfer volumes, mixing, shaking, incubation times, they're all done the same way every time.
Okay, but then I got two questions for you. So what if the customers don't want to do the same thing every time? They want to run a different number of samples, start or stop at different points, change the number of PCR cycles. And then second, don't you guys have to spend a lot of time getting the liquid handler to pipette accurately or mix things well?
Yeah, for the second one first, yes, we did initially have to spend a lot of time working on the pipetting, but because we have so much experience, we can take all these techniques that we've developed in the past and then apply them to our current development projects, and it really speeds up the whole development process. And then to your first question about having variable numbers of samples or start-stop points, just like the pipetting experience that we built over time. We've created everything from completely locked down methods to methods that give users a huge amount of control over all the settings in the method. What we found is somewhere in the middle is really the best spot to be. We know that every lab's process is a little bit different, but what we need to do is allow the right parts of the workflow to have the appropriate amount of control. But this is the other big thing that's underestimated. Building in this flexibility is not only tricky, but it creates a lot of paths that a method can take, each of which have to be then verified. So this adds a lot of time to the development process and a lot of expense to it. So actually, Teri, we do have a brand new solution called the AVENIO Edge. That's a great example of everything we've been talking about here. Andy, could you share some information about the new system?
Thank you both for sharing your experiences, Teri and Scott. I'm sure many folks in the audience can certainly relate to your stories and experience. Doing NGS sample prep manually is a way in the past, to be honest. But automating NGS workflows is all too often a privilege of labs that have the automation expertise. It takes a lot of time to train new users, implement long and complex workflows that involves many device integrations, and not to mention work with multiple vendors sometimes, while under the pressures to meet all the project timelines can be very tough. And this is why we're launching a new product called the AVENIO Edge system. The AVENIO Edge system is a Class 1 IVD system that takes purified DNA and prepares the sequencing-ready libraries. We launched this product earlier this year, and it is now commercially available in the US. It's fully enclosed and integrated with all the necessary devices needed for library preparation, target enrichment, post-capture pooling, and library quantification, all in one box. AVENIO Edge is quite unique in its kind because it provides what we call a true walkaway experience to its users. As we know, many automated workflows still requires the users to be around and frequently come back to the system. Sometimes that's because not all the devices are fully integrated with the automated liquid handler. Sometimes certain steps just require a little bit more of babysitting, right? Making sure the liquid handler is doing what it's supposed to do. On the AVENIO Edge system, for each sample run, only 20 minutes of manual setup is needed. After the run starts, the user can just simply walk away completely and work on other things without constantly thinking about when and whether there's the need to go back to the workbench. We have a short video that showcases some of the key highlights of the AVENIO Edge system, and I'm going to play that video right now, and we'll come back to discuss more about these features. All right, as you saw in the video, we designed AVENIO Edge to specifically address those challenges around the traditional NGS automation solutions. Scott, would you please take a moment and share more about these features that we just saw in the video?
Yes, Andy, I'd be happy to. Full integration of a thermocycler, heater shaker, cooling blocks, a proprietary magnet position, and an onboard quantification module are the key features that make this a truly walkaway system. One power button turns the entire system on, including all the integrated devices I just mentioned. Based on the number of samples selected for a run, the integrated glove-compatible touchscreen provides step-by-step placement instructions for each consumable. To ease instrument setup, the tip and reagent areas of the deck are on slide-out drawers, and the instrument has bright LED lighting. Reagents are ready mixed and barcoded. Preparing and dispensing master mixes is no longer required. All the plastic consumables are barcoded. Pipette tip racks are barcoded as well as color-coded, and partially used tip racks can be left on the instrument and used in the next run. At the start of each run, the system first scans all the barcodes to verify the deck setup is correct, and then performs an additional physical verification utilizing the pressure-sensitive capabilities of its SPAN8 head to verify things like the microfuge tube caps are removed, and then we'll also use its gripper to verify the plates are not inverted and a barcode was unable to be scanned. Finally, the built-in control software allows users to customize run parameters such as amplification cycles or conditions, ligation and hybridization times, and bead cleanup ratios. The software also provides a complete log of the run for LIMS integration, and it also records weekly maintenance and has an integrated UV decontamination option.
Thank you so much, Scott. To show how AVENIO Edge can truly transform users' experience, we actually performed an internal study to compare the AVENIO Edge HyperCap workflow to our manual Kappa HyperCap workflow, as well as the same workflow on 2 other open platform automation systems. As you can see here on this graph, AVENIO Edge only requires 14 manual steps compared to the other workflows. And this includes the normalization of input samples, DNA library prep, target enrichment using our Kappa hyperexome panel, library quantification, and post-capture pooling. As a result, as you can see on the right here, AVENIO Edge significantly reduces the manual steps required for this workflow. And the opportunities for user errors are also reduced by 91% compared to the manual preparation and by 88% compared to the other 2 automation workflows. In the same study, we also looked at the hands-on time required to complete this workflow. Again, this includes input sample normalization, library prep, target enrichment, library quant, post-capture pooling, as well as the time required to clean up the workstation after the sample run. The graph here shows AVENIO Edge only requires 106 minutes of total hands-on time. And as you can see here, doing all that manually or on the other automation platforms require a lot more time. In fact, AVENIO Edge can help save over 9 hours of hands-on time compared to manual preparation. And what's also important here is that these hours of time saved are uninterrupted. So AVENIO Edge is a system designed specifically to offer a refreshing NGS automation experience. In addition to the product design, we also want to leverage Roche's experienced and extensive support network to offer our users a peace of mind. For example, AVENIO Edge instrument and all its integrated devices, software, applications, and even reagents are all supported by Roche. This means an AVENIO Edge user would no longer need to work and coordinate with multiple different vendors. The deployment or implementation of AVENIO Edge is also much faster compared to the other traditional automation systems. Installation takes about 3 days. There's no additional integration services needed because it comes with all the devices already integrated. Our field application scientist would also provide basic user training on site. No prior automation experience and knowledge is required. And this means potentially it can get all set up and running within just a couple of weeks. And also the accuracy of pipetting obviously is very important during the NGS sample prep process. Our Roche service team will ensure the entire system, including all the devices, are calibrated to their optimal conditions. Pipetting verification is performed during every AVENIO Edge installation and preventive maintenance service. The routine user maintenance is also very simple, and there's an on-screen instruction for the user to follow very easily. So we believe AVENIO Edge system will be able to help many labs transitioning away from manual NGS and overcome the traditional challenges with lab automation. If you have any questions about AVENIO Edge and what we talked about today, please visit sequencing.roche.com/AVENIO Edge. Simply fill out a short form and we will be more than happy to further discuss with you. We also have a self-guided virtual tour available for AVENIO Edge if you just want to check it out and learn more about these features we talked about today. With that, this concludes our session today. Teri and Scott, I want to thank you both again for joining me and sharing your experience with us. To all of you tuning in, thank you so much. We appreciate your time and interest, and have a great day.
Thank you, Teri, Scott, and Andy, for your time today and your important research. Before we go, I'd like to thank the audience for joining us today. I'd also like to thank our sponsor, Roche Sequencing, for sponsoring today's webinar. Questions submitted today and during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand for 2 years until May 18th of 2024. Labs will alert you via email when it's available for replay. And we encourage you to share that email with any of your colleagues who may have missed today's live event. Until next time, goodbye.